Hello, everyone. I would like to thank the organizers for granting me this opportunity to present my research at GLBIO. I also thank all of you for your interest in my work. Today, I will talk about a eukaryotic gene annotation pipeline that I developed in collaboration with USDA scientists. Before I delve into Finder, I would like to give you a brief overview of the problem that we are trying to address here. Gene annotation is the task of locating transcriptionally active regions of the genome and building complete gene structures with their constituent exons and introns. In addition to that, gene annotation also entails correctly detecting transcripts. For example, this region of the ZRMA's genome houses three transcripts of a gene. They differ from one another by the number of exons and in the coding regions. Several eukaryotic genes contain an alternative arrangement of exons and introns. During transcription, both introns and extrons are transcribed. Matured mRNA is formed from the pre-mRNA by eliminating the introns and merging the contiguous exons together. It is this matured mRNA that is sheared into small fragments and are prepared for sequencing. The sequenced short reads are mapped back to the genome to locate the regions of transcription, and we use that information to construct the genes and transcripts. Currently, there are multiple approaches to annotate genomes. Those could be broadly divided into two categories: prediction based and expression based. Breaker 2 takes a completely prediction based approach, while PASA often utilizes expression based evidence like RNA-seq or full length sequences. Maker is designed to use both prediction based and expression based data, but it is not completely automated. Unlike Breaker and PASA, Maker 2 must be executed for multiple rounds and is heavily weighted to favor predictions. To overcome all these drawbacks, we have designed Finder, which is completely automatic. Finder can download data from NCBI SRA. It can align, assemble, and annotate with no manual intervention whatsoever. Finder reports a mapping between transcripts and tissues, which could be later utilized to detect tissue specific transcripts. Finder is optimized to utilize gene expression data from RNA-seq datasets. It predicts gene models using ab initio predictors and can use protein sequences to further improve gene annotation. I will briefly go over the pipeline that Finder implements. Finder starts by downloading raw RNA-seq data from NCBI and then aligns it to the genome using star and oligo, two aligners. Then these aligned files are fed to the assembler Cyclas, which is a meta-assembler. So it produces a consensus assembly in addition to individual assemblies for individual RNA-seq samples. So we looked at these consensus assembly and we found that there are certain gene structures which are incorrectly annotated. So we modify those gene models, and I'm going to talk about this modification and how we do it in a future slide. And we obtain an enhanced assembly. Then this enhanced assembly is sent to another program called GeneMark-ST, which is used to predict the coding region within a gene. So that gives us a file with coding sequence annotated. So RNA-seq expression data is great for constructing genes that are expressed. In order to capture most genes, one need to analyze data from very large number of RNA-seq samples collected from a variety of tissue types and experimental conditions, which is difficult to obtain for several reasons. Hence, Finder uses gene prediction pipeline to capture those gene models that were not found in the RNA-seq data. Users have the option of providing protein sequences in FASTA format. Finder also aligns these proteins to the genome to generate annotation. Finder combines the non-overlapping gene models constructed from these three sources and then assigns confidence classes to them. So if a particular gene is found in both RNA-seq sample and in protein, it is given a higher confidence. And if a particular gene is found only only as a protein and there is no other evidence, then it is assigned a lower class confidence. So Finder uses a single assembler to generate transcriptome assemblies. So it was shown in this paper that the meta-assembly approach undertaken by Cyclas worked better than string time merge or TECO. So in this particular diagram, you can see with increasing sample size, both precision and recall for for Cyclas is almost same. But if you consider the other consensus assemblers like string tie merge and and scallop plus teco, you will find that some of them for some of them the recall or the precision it reduces as the sample size increase. So in addition to Cyclas, there are other softwares that perform meta assembly of the RNA-seq data. So these programs accept annotations constructed from individual RNA-seq samples and then generate a consensus assembly. We compared Finder with the current softwares and we found that Finder was able to produce the best results. So we developed Finder on Arabidopsis thaliana since it was easier for us to track errors in gene annotation with its almost perfect gene models. One of the biggest challenges in gene annotation is to correctly annotate adjacent genes that overlap with one another and reside on the same strand. Short reads that originate from the ends of these genes overlap with one another. So in figure 1a, Cyclas was unable to detect two different genes and instead reported them as a single one. Finder uses statistical change point detection to split these genes based on read coverage. So change point detection is used when there is a statistically significant read coverage trough in one or more of its internal exons. Change point detection is also used to modify the boundaries of overlapping genes on the opposite strand as depicted in this figure. So we generated gene annotations for Arabidopsis thaliana with Finder and compared it to the ground truth models. We used a multitude of different metrics for comparison, but I'm going to discuss only a few here. One of the metrics we used to assess the quality of Finder gene models was annotation edit distance. So each transcript in the set of annotation can be assigned an annotation edit distance that indicates how well an equivalent annotation exists in the reference. A higher number of transcripts with lower AED scores is indicative of a good annotation. So we applied Wilcoxon's sign rank test to establish that the AED score of Finder gene models was significantly lower than other approaches. And this is further confirmed by the figure on the right, which reports a larger percentage of transcripts belonging to lower AED scores in Finder as compared to other annotators. For more details about the analysis, I welcome you to check out our publication in BMC Bioinformatics through the QR code. You can also check out our software, which is hosted in GitHub. So in addition to AED score, we use specificity and sensitivity of the annotation to judge its quality. So it is almost impossible to detect more correct transcripts without constructing a few false positives. Hence, we computed the F1 score that encompasses both specificity and sensitivity. A multi-exonic transcript is deemed correct if the entire intron chain matches perfectly with at least one reference annotation. So for monoexonic transcripts, a match of 80% with a reference sequence transcript is required for it to be recognized. Even though some annotators perform well at the base and the exon level, they were incapable of forming correct transcripts, leading to a low F1 transcript score. So here we are more interested about the transcript score because transcripts encompass both base and exons and is the actual biological entity that others will later use. So we categorize these genes based on different criteria to different groups. So there was a group with uni-exon transcripts. So this contained only those genes which had one transcript. And there was a group of transcripts without any UTRs, then groups of transcript with UTRs, and other such similar kinds of gene groups. The hash symbol indicates the performance of the particular annotator which did best in that particular group. So if you look at ZMAs, you'll see that in ZMAs, almost all, in fact, in all the groups, Finder was able to perform better. It generated much better gene annotations. In case of Arabidopsis thaliana, in most of the cases, Finder did really good. But in one or few cases, Breaker did better. Like the case with transcripts without UTRs and uni-exon transcripts or genes with a single transcript, there it did a little it did a little worse. So the main reason is behind how Breaker was created. So the breaker annotates only the CDS region of the genes, and hence it is expected that it will perform better with transcripts without UTRs. Now we actually inspected these transcripts without UTRs using some PacBio data, and we found that there was evidence of UTR present for those genes also, but they were simply not annotated in the ground truth. So even with Arabidopsis, there were some missing UTR annotation. So next we classify the genes according to the similarity to genes in other species. So genomic phylostratigraphy is a method to date the origin of specific genes by looking at its homologs across species. Cellular organisms is the clade that contains the most conserved genes, and whereas orphan genes are housed in the rightmost in the topmost clade. Genes are exclusively assigned to each of the clade. In the figures, we have shown only those clades which have more than 500 genes. Finder was able to detect more transcripts correctly for all the evolutionary clades, which is a little surprising because um, Breaker works with homologous proteins and should have been able to, do well in the cellular organisms clade, but apparently it seems like it was not able to do very well. So this analysis was done by our co-author Priyanka Bhandari from Dr. Eve Wortley's lab. This brings me to the end of my talk, and I would like to summarize some key points. To leverage the huge reservoir of RNA-seq data, we offer Finder, which is entirely automated, making it perfect for bench researchers. Finder combines the ab initio gene predictions with gene models constructed from RNA-seq evidence, incorporating the best of both approaches. Compared to other gene annotation softwares, it is much, much easy to install. And currently, we are working on a method to generate the consensus assembly from individual assemblies, which will offer the opportunity to update existing gene annotations with novel RNA-seq data. With that, I would like to acknowledge my committee members, my former lab members, friends and family, mentors, lab members, and all my funding sources. Thank you. I would be happy to take any questions.